All right, good morning, everyone. We're just going to start with a couple of announcements. First, for anyone that's new, which I think I see some people have never been, I haven't seen before on the call. This is kind of a regular every other week type seminar, all on Zoom so that we can be joined by everyone from around the world. And we have a mailing list, which is this top link here. So please add yourself to that if you want to get announcements about the seminar or other events happening. For a lot of talks, we also record them and post them onto this YouTube channel. So you can watch past talks. And please put in any speaker recommendations, either email me or Magnus about speaker recommendations, either yourself or someone else, particularly interested in trainees, early career, and underrepresented scientists. And just to note, we do have a slot open for the fall still, 5 December. So if anyone is available for that time and wants to have a talk about some aspect of RNA 3D structure, please let us know. The next meeting will be in 2 weeks, same time, and Jan Ying will be speaking about TR Rosetta. All right, so for today, I'm going to introduce Rhiju Das, who will be talking to us about a big data set that has been coming out of the lab. So Rhiju initially trained in physics, both at Harvard and UCL, and then came to Stanford to do his PhD in kind of RNA structure, ion composition around RNAs, etc. He then went to more the computational folding side of things in the Baker lab, before returning to Stanford to start his lab, which is focused on modeling RNA 3D structures computationally, as well as increasingly getting experimental data representing RNA structure. So we're very excited to have him today speak about a new data set coming from the lab that he's very excited to share with the community. All right, Rhiju, I'm going to unshare my slides, and then you can get started.
Sure. Thanks, Rachel. Thanks, Rachel and Magnus, for organizing this series. And thanks for all of you for coming. Let me get this going. Before I start, I want to extend my sympathy to all of our colleagues and their families who are in Israel and Palestine. And so yeah, our thoughts are with you. And this war is a terrible thing, and we hope it ends soon. We're very fortunate to be able to come together and talk about our favorite molecule here. So I hope we can still enjoy it. As some of you know me, my dream for a very long time, actually, since I started my graduate work at Stanford, has been to be able to take any RNA molecule that I might read about in a journal or discover myself or design to be part of let's say, an RNA therapeutic, and to be able to quickly model whether it has a three-dimensional structure, and if so, what is that structure or set of structures. And I'm showing you here an example that some of us know really well. This is a model of the tetrahymenorybozyme, the first ever RNA-only enzyme discovered by Tom Check and colleagues in the early 80s. This model actually came from Eric Westhoff and his colleagues in the 90s, and it has guided hundreds or maybe thousands of deep experiments in understanding RNA structure and function. And it turned out to be reasonably accurate. So in 2021, not too long ago, several groups, including mine with Rhiju, helped solve the cryo-EM structure of this RNA. And the global fold of Eric Westhoff's model turned out to be quite accurate. And it would be wonderful to be able to get these kinds of models for the many millions or maybe billions of RNA domains where we suspect their 3D structures might be important for their function. In other talks in this series, you'll hear about the RNA puzzles, community-wide blind predictions that that has driven progress over the last roughly 12 years or so. But a very exciting event, and one which I think has brought many of us to this session, is CASP 15, where we had the first RNA category. And there I wasn't a predictor, but helped assess this category along with Rachel, Xichao Miao, and Eric Westhoff, and then several other colleagues from the protein structure prediction community. And we were really excited to find that for many of the targets, there were 12, there were quite good models. So these are 4 targets that were solved by X-ray crystallography. These are very interesting molecules, including the first RNA-only ribozymes that are known in humans. And in blue is the best model from CASP, and in green is the experimental model. If I move forward, I'm going to show you most of the targets that came from CASP. I'm going to, I just need to do a little cleanup here. Okay. Most of the targets that we had in CASP came from cryo-EM. These included design nanostructures from Ewan McRae, Abby Anderson, and others. Those are shown here on the right, as well as segments of coronavirus genomes that actually Rachel and I and Rhiju and others have solved. And here again, I would say the models were visually quite excellent. The only really difficult cases seem to be RNA protein complexes. Here, 2 RNAs that act kind of as sponges for a bacterial riboregulator protein, and this is solved by Xiaoming Su, and there weren't I would say, any good models. But for 10 out of these 12 cases, there was I think pretty outstanding success, and a success that we maybe didn't expect, particularly for these very large nanostructures, for which the RNA puzzles competitions hadn't had examples before. But there was also a big surprise, which many of you know, when we ranked the different groups. The majority of these really impressive models came from 4 groups, which I'm highlighting here on the top. And we had assumed, based on the preprints that had been coming out throughout 2021, that these groups would have been leveraging deep learning approaches analogous to what had been pioneered by Google DeepMind with AlphaFold for proteins. But then when we looked at the abstracts of the folks who were, when they described their methods for the CASP conference, the ones that use deep learning or some variant of it are highlighted here in orange. And actually the top 4 groups did not seem to use deep learning. And now you may know that these top 4 groups have written their own papers. They're all out in the special issue of Proteins, and they confirmed that they didn't use artificial neural networks. And so I think for a lot of us, this is, on the one hand, nice, but we'd like to get to a point where we don't have to rely on Hongxiang or Shiji Chen or Martha's group or Janusz's group to help us solve structures. Absolutely, because that doesn't scale. You know, if you're going to try to get millions or billions of RNA domains, you need to have something automated, and something kind of like AlphaFold. And so how do we get there? That's been kind of the big question, I think, certainly for me, and I think for a lot of us in the field. Well, you know, for other areas where deep learning has worked, you know, whether that's for protein structure prediction or modeling language or images or video, you need 3 ingredients. You need models that can be trained, you need some form of rigorous evaluation, and you need typically big data sets for training. And as you can see, we actually have now several models that can be trained. Most of those groups that were highlighted in orange in the previous slide have made their code open source. And so that's a wonderful thing. We have something like 18 to 20 models that we can play around with. We have rigorous evaluation in the form of the RNA puzzles and the CASP trials. And what it seems like we don't quite have are the data sets for training. In particular, our crystallographic and cryo-EM databases of RNA structures, although they're growing, still have probably no more than, let's say, a few hundred non-redundant structures. And that just doesn't seem like enough for training a deep learning algorithm. AlphaFold required something like tens of thousands, depending on how you count it, to hundreds of thousands of structures in the protein crystallographic data bank. So how do we expand our structural data sets from 1,000-ish RNA molecules to 1 million RNA molecules? That seemed like that might be enough. So that's what has been the focus of me and several folks in my lab. basically ever since the CASP conference last December. And what I'm going to tell you about today is a snapshot of our progress in that area of getting big data for RNA structure inference. And so our plan is, okay, well, first of all, to get structural data on 1 million sequences, you kind of have to have 1 million interesting, diverse sequences. It can't just be tRNA over and over again, or the ribosome over and over again. And so, we're fortunate to have these databases like the Protein Data Bank, but also RFAM, which are in some ways crowdsourced databases of interesting RNA molecules that have been crowdsourced to the expert RNA community. We also have, large databases of genome sequences of viruses and every other living organism. Although it is kind of a challenge to figure out what domains there might have interesting structures. And I'm going to tell you a little bit more about another source of structures called Eterna, which actually has ended up being the most powerful source for this initiative. Once you have those sequences, then you need to get a lot of structural data. And we have access to cryo-EM and other 3D structural biology techniques, but I'm going to tell you a little bit in the talk about another approach called chemical mapping, which is, I'd say, uniquely powerful for RNA structural information. And it also has its limitations. And then the idea is that if you got all these structural data, that might be enough, well, minimally to rank the existing methods. And we have really a zoo of methods, particularly for secondary structure. I'm listing a few here, where we have an emerging set of 3D structural methods, including the ones that have arisen as part of or in parallel with the CASP 15 work. And I'm, again, just listing a subset of those here. So you can imagine that these data would be enough at least to rank the different structures. And perhaps there might be enough here to fine-tune or retrain some of these algorithms, especially the ones that are differentiable neural networks, like the 3 that are listed down here. And then if that isn't enough, then hopefully you can then use these updated algorithms to go and search genomes or to look for other families of interesting RNAs. And maybe you'll get another 1 million, and then you can go through the cycle again. So that was the hope. And I'm going to tell you about some progress in each of these steps. So I'm going to start with the sources of interesting sequences. And it turns out that kind of like in other areas of machine learning, just like ChatGPT is trained on lots and lots of sentences originally from the internet and now from people who are using a chatbot, it's a crowdsourced data set. We reasoned that the best source of diverse RNA sequences could also come from a crowdsourcing approach, an internet scale, non-expert crowdsourcing approach. And they were fortunate to be part of a project called Eterna, which has been crowdsourcing RNA design now for almost 13 years. So Eterna is this video game project. The URL's in the bottom. You can also get this on your phone through iOS or Android. Eterna's tagline is "Played by humans, scored by nature." And since its launch in 2011, we've been putting out puzzles to design interesting RNA structures and functions in the form of this mildly addictive video game puzzle, which challenges users who don't have to have any prior experience in RNA to return sequences of blue, yellow, red, green symbols that correspond to actual solutions to these RNA design problems. And then this community, they publish these sequences, they vote on which ones they like best, and we take the top ranked sequences, or sometimes all the sequences, and synthesize them in our lab. And then we and our colleagues have invented high throughput methods that let us characterize whether they have the structure or function that we ask for, and then those data go back into the video game for the next round of puzzles or the next problem. And through this virtuous cycle of video game playing and wet lab feedback, Eterna has now put out over 30 peer-reviewed manuscripts, many of which include players as co-authors. And in fact, several of them have been written by the players themselves or have been written entirely by the players themselves. So this is a tremendous source of in some ways non-expert, but highly well-trained knowledge about RNA. And this has resulted in the most designs for our current effort. The way that we actually solicited them is through a challenge that we call the OpenNot Challenge. This is a screenshot of a splash page for OpenNot round 3. we made available in the game a folding engine that tries to, not very accurately, but tries to model pseudoknots, which are a kind of complex RNA structure that will probably require understanding 3D folding to design correctly. And then we are, in this case, in round 3, we're listing nearly 1 million designs in this round. And then what we've been getting back since about last December are extremely diverse designs. So these are designs where, sorry, this is a t-SNE plot that is showing sequence clusters of these different designs. Some of the players that have produced these designs are marked. And the main thing I want you to see is that these designs are really all over the place. It's not like one sequence, or it's not like one tRNA, which would form its own cluster, dominates the plot. There's really lots and lots of different things. So now that we have the source of designs, and it's really getting to the millions of scale, we have to find a way to structurally characterize them. And so that'll take us to this second part of the talk. And by the way, I only have half an hour of slides, so or maybe less. And so if people want to chime in with questions, then hopefully Rachel or Magnus can read them out to me. Okay, so in terms of getting structural data, on the one, like I said, we have abilities now to get three-dimensional structures for for many different RNA sequences. A few years ago, I was involved in helping create this method called ribosolve, which brings together cryo-liquid microscopy, as well as something called chemical mapping. And that's accelerating our ability to solve 3D RNA structures, but it doesn't seem to be fast enough to actually solve 1 million RNA structures. But a key part of this method actually is the chemical mapping, which we need to at least minimally assign the secondary structure of the RNA. So I'm going to explain that to, especially those of you who are coming more from the protein side or the machine learning side, how this technique works. Okay, so this is a slide from a really nice graphic from Julius Lux's lab. Explains the idea. Suppose you have a bunch of RNA.
Before we go into that, because I had the same question and I had to Google. So Katie asked, can you go back to this tsnae plot?
Yeah.
And I Googled, so I know already what it is. But can you explain what is it?
Okay, yeah, right. So
it's not for our community RNA 3D structure prediction. It's not, I think.
Okay, yeah, that's a good idea. So this is t-SNE is basically a tool for it's a dimensionality reduction tool for visualizing data sets. It's not that useful on its own as a modeling tool. It's more, it was actually elicited from a crowdsourcing challenge about 10 years ago. And it was invented by folks from the neural network community, actually before neural networks were hot. And it's a way to take, if you have data, like in this case, we have sequences, a whole bunch of different sequences, but in other cases, it may be coordinates of a protein, or it could be a bunch of frequencies of words in text documents or whatever you like. Then those instances live in a big multidimensional space. So an RNA sequence, even if it's just 100 mer, lives in something like a kind of like a 400-dimensional space. So t-SNE is a way to try to pull, it's one of several methods that can try to create a 2-dimensional map or a 3-dimensional map of those sequences, where a human being can then go and assess how clustered is it, are there interesting subclusters or other patterns. And so t-SNE, I forgot what it stands for. I think it's stochastic neural embedding, but I could be wrong. And the T refers to a special way of
T-distributed stochastic neighbor.
Yeah, it's just a way to change the distances to be a little bit more, kind of like a z-score, a little bit more normalized. And then it turns out to be extremely useful. This and UMAP are probably the most common ways that we see for visualizing data in the machine learning communities. And I thank you for asking the question. Okay, so I'll come back to now to experiments. How do we actually, structurally characterize all those sequences? So this technique has been around for a long time, and this is just a nice little graphic that came from Julius Lux's lab. So imagine you have an RNA or a set of RNAs. There could be the red RNA, the blue RNA, the green RNA, and they have different structures and sequences. Then what this method doesn't give you the 3D coordinates of those RNAs, much less the 3D coordinate ensembles of those RNAs, which is what you want. Instead, it relies on a observation that was made, I think in the 1970s by Walter Gilbert and colleagues, that if you treat an RNA with a certain chemicals, one is called dimethyl sulfate, which methylates, for example, A's and C's. Another set of chemicals are reagents that acylate the 2' hydroxyl. And it seems kind of esoteric, but what these do is they seem to only tag the parts of the RNA that are in loopy regions. And so if an RNA is forming like a little helix here, then the base pairs there somehow prevent chemical modification by those chemicals. And so you treat this RNA sample just with that chemical, you get these marks that show up on the RNA, and then there's a bunch of tricks that allow us to detect where those marks were. What we're going to be using in this talk is we'll be reverse transcribing the RNA to convert to DNA, and the reason for that is because we have these powerful high throughput approaches based on DNA sequencing. And then there was a trick that was this, I think, first reported by Kevin Weeks' lab in North Carolina, where several reverse transcriptases, when they try to make a copy of the RNA to DNA and they hit one of these marks, they will either stop, or in some cases, they'll actually bypass that mark. They won't know what nucleotide to put, so they'll put in a mismatch or a deletion, and then they'll just keep going all the way to the end. And so by reading out the cDNA and looking for those mistakes, we get an estimate, we get a good picture of where those marks were in the original RNA. And then we get these kinds of, for example, bar graphs where, like for the green RNA, there are these segments here that are protected. Those are likely forming base pairs or other kinds of structure. And then there's other parts of the RNA that are highly reactive, highly modified. And so those are likely forming loops. Okay, so that's not that much information. It's certainly not enough to solve all the coordinates. And so you might wonder, well, why, like, how could training from these data help you in getting to a better model of 3D structure? And here, so my lab and I have been working on this for a few, these kinds of approaches for a few years. And one thing that I think gave me some comfort in moving forward with chemical mapping was this idea that chemical mapping, although it seems like there's not that much information coming out of it, it's just one number per nucleotide, there's ways to hack it to get much more information about RNA structure. And so I'll describe one of these hacks in this slide. Imagine you have an RNA. This happens to be data for a bacterial RNA that senses adenine. It's called an adenine riboswitch. And I'm showing you here the actual data that we get from chemical mapping. And in here, black is a nucleotide that's highly reactive. And then gray or white these bands correspond to nucleotides that are not that reactive. So they're probably forming a base pair. And so if you concentrate here on my pointer where it says U28, that U is probably forming a base pair with something else in the RNA. Okay, now these data alone don't tell you what that partner is. It could be A21, it could be G42, it could be A52, you don't know. Okay, but the idea here is that suppose you made a mutation of U28 like to an A. Okay, and then you created that RNA and you got the chemical mapping data. And suppose in that RNA, there's one other nucleotide that became reactive, G42. Then you could infer potentially that they were originally base pairing in the wild type sequence. So here's a little pictorial. Imagine that there was a part of the RNA that looked like it formed into a hairpin, and then you made a mutation here, and another part got exposed. Then this tells you that they were likely base pairing in the original one, and now they form a mismatch that is reactive on both sides. Okay, so what we have worked out over the last few years are ways to actually make all these single mutants. And I'll show you an example of that actually for that adenine riboswitch. Here's the wild type profile for chemical mapping. And each of these bands is a different nucleotide. And then I'll show you, going down the screen, what happens as you start marching along mutations from the 5' end to the 3' end of the RNA. And so I hope you can see that there's like a kind of a diagonal feature here. So that's where if you make mutations, experimentally near those sites of mutations, there's often a disruption in structure. But then what's really useful is to see that sometimes I'll make a mutation here at whatever it is, U20, and then I'll get a mutation somewhere else, in this case later in the sequence. And those are likely going to be base pairs, okay? And then we have automated ways to go basically calculate z-scores to try to figure out what might be base pairing. And it turns out that also shown here in purple, you will sometimes get signals here for not just Watson-Crick-Franklin base pairs, which help you read out secondary structure, but sometimes you'll get signals that correspond to non-canonical base pairs that are the hallmark of tertiary structures as well. And what's shown here in purple happen to be some of those non-canonical pairs. And so in our early work, we showed that you could recover RNA structures that have no structure, like this adenine riboswitch from these kinds of data. And then within the puzzles trials, actually for something like 10 of those puzzles, we actually we call them Mutate-and-Map data and made them available to everyone who's making predictions. And then that led to increases in accuracy in secondary structure and tertiary structure modeling. And there's all sorts of additions that involve making single mutations like here, or double mutations, or even up to 3 or 4 layers of mutations that let you infer properties of an RNA's structure, as well as its ensemble of structures if it forms multiple structures. Now, I'm not going to go into detail there. The key point that I want to make is that you had something, in this case, an experiment, but suppose you had a computational oracle that could give you back chemical mapping data. It would have to understand a lot about RNA structure. Okay, so that's the key point. If you can feed something sequence and get back the chemical mapping pattern, it's got to understand, and if it's perfectly accurate, it's got to understand a lot about RNA structure. So how are we going to actually, well, so we have 1 million sequences. We have an approach that's quite sensitive to RNA structure, an experimental approach. Can we scale these chemical mapping approaches to 1 million arbitrary RNAs? And again, just because of the audience of this talk, I'm not going to go into all the nitty-gritty of how we did the scale up. It turns out it's not that hard. Several really nice innovations just in the last 2 or 3 years from Danny Incarnato's lab, Tony Musto's lab, Anna Pyle's lab, have given us chemical reagents that produce high signal to noise. And I'm showing you here data on, these are just a few hundred Eterna sequences that came in, I guess, last December. And then actually, I went back in the lab and figured out how to get high signal to noise data. Red here means that each row here is a different RNA. Red means that it's reactive, white means it's not reactive. In one case, the reactivity pattern on the modeled secondary structure of the RNA, this appears to be a pseudoknot. And there are some additional tricks here for the aficionados. We end up putting unique barcode sequences at the 3' ends of the RNAs, which help us deconvolve the data at the end from the sequencing runs. Maybe the major upshot, I think, particularly for those of you who are doing these, I think a subset of you who are actually doing these experiments in your labs, is that on the one hand, in our pilot experiments, we're looking at 10,000 profiles, and it costs quite a bit of money. It costs about $50,000. But it turns out most of that cost is our own time, our own personnel time. The costs of sequencing and the cost of synthesis have been going down dramatically. And so we realized that if you wanted to get a million profiles, that would require an investment of not 100 times more money, but something like 3 or 4 times the amount of dollars. And that's, although it's expensive, it's feasible, and we can budget for it. And we decided to invest our lab budget and our lab time into getting 1 million profiles. So let me just show you what we discovered in the initial pilot from these chemical mapping data. We took a bunch of Eterna designs, and then we have ranked them here based on how consistent that their chemical mapping profiles are with them being pseudoknots versus not, okay? And I'm not going to go into, again, details about how we create a score from 0 to 100 to rank them. But I just wanted to show you like what sequences there are, because this also gives you a sense for how diverse these RNAs are in this data set. So the top sequences here were submitted by a player named Digital Embrace. And she had actually taken sequences that were previously considered in Eterna for a different puzzle that was led by Cody Geary, who some of you know is one of the co-inventors of RNA origami. And so this is actually a little rationally designed RNA that is supposed to be forming in 3 dimensions. And it turns out that the chemical mapping data for this RNA are extremely consistent with it forming this 3-dimensional structure, including kissing loops that help pin it together into a well-defined structure. But there are lots of other sources of RNA. So it turns out that in this top 10, I think 5 or 6 of these RNAs happen to all come from a RNA that was listed in a database called the pseudobase, a database of pseudoknots. And it had come from a paper from the 90s that had isolated through in vitro selection, a pseudoknot that binds the human nerve growth factor protein. And then there's no structure known of this RNA, but here these chemical mapping data are strongly suggesting that it forms a pseudoknot. But not, okay, so these are coming from kind of existing databases, arguably from expert labs. But it turns out then, as you start going down the list, the majority of the designs start coming from totally novel RNA sequences. This is from a player named SP Vincent. This is from a player named Jester. And these sequences, to our knowledge, don't seem to have any relation to existing natural or in vitro selected or designed RNAs. And we don't actually know their three-dimensional structure, but the data that we're collecting, this is actually the in-game data browser, are much more consistent with a pseudoknotted or interesting three-dimensional structure than an RNA that does not form an interesting structure. And if you'd like to peruse these, here's a link at the bottom of the game, or if you just Google Open Knot Challenge, you'll get examples of what these sequences and some of these data look like. And so we have so many data here. The plurality of the sequences come from Eterna, but we also have sequences that come from, we have a representative from each RFAM family of 4,000 RFAM families. We have every sequence in the PDB is in this data set now. Although in some cases we've had to window those data sets. We have many windows of the coronaviruses, although we were careful to only make 2/3 of any virus to ensure that we're still safe in the lab. And so we're in a situation now, and we call this the Ribonanza data set, which is also the name of this, the title of this talk. And we have a little over 1 million RNA sequences. For the aficionados, we have chemical mapping profiles based on a shape methodology, a 2' hydroxyl tagging methodology, as well as dimethyl sulfate or DMS. And the lengths here range from 115 to 207 nucleotides. And then what's in process are a little over 1 million other RNA sequences where we're collecting those profiles. And the lengths there go from 177 to 457 nucleotides. And so in this more latest run, we now have entire group 1 introns, like what I showed in the first slide of this talk. And we have pieces of entire pieces of ribosomes, but we also have really kind of elaborate origamis and synthetic RNA structures going up to the size range. And the other thing I want to point out is we intentionally have kept these later stages to have a different length distribution than what we have in the early distribution, the stuff that's in hand. So that if we develop models based on the data that are in hand, those are our training data, we are going to be requiring generalization to longer lengths in our test data, which are the data that we're collecting now. Yeah, Magnus.
Okay. So yeah, this is really interesting because I don't know if you remember, but when I worked with you, we had this, I think, discussion and idea, and I wanted to ask if you have data for that. So we discussed the alignments, different alignments, and what would be, is it possible to model different RNAs for given family and to understand given, what would be structured for given extra helix or something is missing? And I think we discussed briefly, this kind of shape experiments for the family. So is it in your data set, would you have like design or sequences for the same family but different sequences? And what was the way how you picked these different sequences, if they are there?
There's a little over 4,000 RFAM families, and yeah, so we have multiple members of all those families. After I forgot how I clustered it, so I think at 90% sequence identity. So for some of the families, we have many sequences, and for some of them, we only have one because RFAM is actually not very diverse. The individual families actually are not that diverse, but yeah, so we have those data as well. Yeah, those are interesting for a lot of reasons. One is like to help potentially, if we wanted to get models of those RFAM families, it's nice to have those data, particularly for multiple sequences that hopefully have a similar global fold. And then from the point of view of training and test for machine learning, I mean, it just seems like those sequences are out there already. They're going to be used anyway for all of us who are trying to train machine learning models. So we might as well have chemical mapping to go with those sequences.
Yeah, but it's really cool because then you can look at given family, and if you have a few, a few or whatever 10, chemical mapping, then we could do some modeling and look at the evolution and this kind of like this is a very different. I didn't think about it before, but then when you showed this slide, that would be something really cool that I think I would like to look at. Yeah,
there's a lot you can do, and there's a million. I don't know whether that seems like a lot or a little, but it's certainly more than, kind of what's in RFAM. So so I think, you know, it was kind of a no-brainer to include all of RFM. I should say we only included RFM stuff from the seed sequences and not from the extended set of sequences that come from an RFM search. So that's something that we could do in the future, to be straightforward.
Okay, thank you.
I can, that was, I'm happy to talk about other aspects of the data now also. Or we can come back to it. Okay, so that's how we're getting the structural data. It's this chemical mapping approach. It's this sort of funny one-dimensional approach. It doesn't give you all the data you need to look at RNA structure, but it seems it should be quite sensitive to structure. And another way to say it is if you have a model that makes a bunch of wrong predictions for these Eterna designs or for RNA families in RFAM, the data should be able to tell you what's wrong. And it'll give you a little signal for where to go. And so that takes us to the last step here in the plan, which is to take the data and then to update models. And so as I've kind of alluded to, there's a lot of these models out there. And although I would love it to, you know, personally download all of them and then figure out how to retrain them, and, you know, and somehow get all the compute that's required to do all of this, it's well outside the bounds of what, you know, a single person or a single group can do. And so the strategy that we're taking here is something that we kind of started exploring about 3 years ago in the context of the pandemic. We were getting data on how RNA molecules degrade. And the motivation was that we have been developing strategies that we hope will let mRNA vaccines become more stable. As some of you know, mRNA vaccines cannot get to most parts of the world right now. And so while it's helped us with dealing with, for example, COVID in places like the US and Europe, you still can get mRNA vaccines in places like India or Africa or South America. And the problem is that RNAs degrade, and so they have to be shipped frozen or freeze-dried. And that is technically very difficult. There's no existing safe frozen cold chain to get these medicines out to the places of the world that need them. And so we were figuring out ways to getting big data sets on how RNAs degrade. And we need to get a whole bunch of models, ideally cutting edge machine learning models. And we got in touch with this platform. It's called Kaggle. Those of you in the RNA community, you may not know about it. But it's a really interesting platform for machine learning contests. And you got, let me just point to a few things here. It was a 3-week competition, but it brought together 1,600 teams. And they tried like every possible machine learning trick you could imagine on the data set that we had. And it's really nicely set up. There's a leaderboard, a running leaderboard on data that you have in hand. And then there's a separate so-called private leaderboard, which is data that are fully set aside that no one knows about. In fact, in our competition, because it was the pandemic, we didn't even have the data in hand to score the competition until the competition closed. And then we closed the competition and we announced the results. But by getting a lot of people onto the problem, you know, we were able to get better models than anything that we were making in-house with our, you know, favorite transformers or whatnot. And so for this Ribonanza data set, we decided to take the same approach. We'll let anyone who wants to come and try out their algorithms or create novel algorithms. So that's the name of the competition. The link is down here. You still have 2 months to go. The deadline is December 7th. It starts September 7th, and then it's a 3-month competition. We have a little over 200 teams already participating. Hopefully we'll get more. This is a snapshot of the leaderboard as of yesterday. Like the last competition, the private leaderboard, meaning the way that we're going to finally score all the competitors, is based on data that we haven't actually collected yet. Those are the data that are in process. So actually, none of us know the answer actually know who's gonna win this competition. But I want to emphasize to those of you who are here on the call, especially those of you coming from the RNA side and or machine learning side, you can download all the training data that we have in hand, okay, and metadata. So for example, if you want to know the title of the sequence from an Eterna player, because you think that might help you reason about what kind of model to use, then you can get those titles of the sequences. You can actually get descriptions from Eterna players. For the RFAM sequences, there's a FASTA file that, again, that includes titles with the RFAM family names and so forth. And we've also put up some example features, so-called base pairing probability estimates from secondary structure packages, if you want to use that, where you're totally free to ignore it. You could download it all once you register. It's a big download, it's 88 gigabytes, or if you're interested in doing stuff on the Kaggle platform, Kaggle provides GPUs and TPUs for training models. And so you can actually do everything you want online. So the other thing I like about Kaggle is it sort of democratizes machine learning, in that all you need is like a Chromebook or just some way to get on the internet to be able to do machine learning. But you can also just download the data if you prefer to do that. And then there's interesting discussions that are happening. And so this is my favorite one. This is from a co-host, Shujun He. I'll highlight again in a little bit. But Shujun has this great post on how to check if your model is generalizing to long sequences. So again, the way the competition set up is we actually don't have data for long sequences. We will be collecting those for the test set for the private leaderboard. But there are certain features, there's certain long sequences that are in the test set, like the tetrahymenorybozyme, actually, which is that first RNA that I described at the beginning of this talk. And we have, as part of the test set, a simulated Mutate-and-Map data set for tetrahymenorybozyme. And so there are certain features that we expect to be there in those data when we collect them. And so we're making that available. And now this thread is really fun because leading competitors are all putting up, their snapshots of their predictions for this little test molecule. And then we're talking about like what is happening, why aren't some models generalizing, and so forth. So it's a really, I think, an interesting way to do community science. Okay, so I'm going to actually stop there and summarize, so we have some more time for questions. I've told you about this challenge called the Ribonanza challenge. It was motivated by the observation about a year ago that there's really no AlphaFold for RNA yet, and the likely bottleneck is probably data. And then to collect a lot of the sequences that we want for a big data set, we've initiated the Eterna OpenNOT challenge, and that's now resulted in many, on the order of millions of diverse molecules. We've then scaled up classic chemical mapping approaches to get on the order of 1 million profiles using these shape and DMS chemical mapping approaches. And then we've launched this competition, and on September 7th, closes December 7th. You still have basically 2 months. That's called the Stanford Ribonanza RNA folding competition. It's on Kaggle, and anyone who wants to participate can participate. And then hopefully, when we get these models in, a condition of winning a prize is that your model available, open source. And so those should all be available in December. And working with some of my collaborators here, we'll be trying to put out a preprint describing how those models work or don't work by early 2024. And hopefully that lets us as a community leverage whatever insights there are, whatever models there are in time for CASP 16, which of course begins next April, 2024. So I'd like to thank the folks who've been doing this work with me. This is a snapshot of several of the folks in my lab. Ray Huang in particular has been carrying out the experiments with me over the last 9 months. Rachel Kresh wasn't in this picture, but she's organizing this seminar series, and also is responsible for the scripts that have allowed us take diverse sources of RNAs and pad them and to get them to the right length, and to barcode them for the Ribonanza data set. I'd like to thank the Eterna developers who have launched this OpenNOT challenge. Advice from several folks I mentioned, Danny and Tony. Joe Yesselman, also from University of Nebraska-Lincoln, has been offering advice. There's a bunch of folks who've helped pilot the machine learning. Like, can you learn anything from these data sets, including a team in a computer science course here at Stanford. We have our collaborators at Kaggle, Walter and Maggie. And then I especially want to thank Shujun He, who's also here in the call. Shujun actually I think, got into the RNA business because of the Kaggle competition 3 years ago, what we call the Open Vaccine Competition. And since then, Shujun has both become a Kaggle Grandmaster and also has been doing his PhD work on RNA. And so we asked him this year to potentially not be a become a co-host to kind of come to the other side and help us set up the competition and lead forum discussions. And it's been really, really to have him on board. And of course, I'd like to thank the Eterna players. This is a subset of the folks who have submitted designs for the OpenNut Challenge. Oh yeah, then I'm going to leave you here with a link to the Kaggle competition. And then, yeah, I'm happy to take questions.
There is one from Yoshi. You want to read it? Or should I read it? If you want. Go ahead. I don't hear you. You're not unmuted, but it doesn't work. If not, then I will just read it. What would you say is a good baseline level of performance on the Ribonanza task? At what level of performance would you say a model is useful in real experiments?
That's a good question. Yeah. What's the useful level of performance? We will be able to read that out because in the test set we have numerous windows of RNAs from natural genomes where we don't know whether or not they have interesting structure. So one downstream task that we really, really care about, we don't care actually too much about that task of chemical mapping in and of itself. I mean, there's maybe the one real world task where that matters is actually in designing RNA medicines that won't degrade. The degradation process is a kind of chemical reaction that's analogous to what we're doing. And so we do care a little bit about that. And probably after Kaggle completes, we'll ask the leading competitors to make predictions for some of these degradation tasks. But the thing that I'm really interested in is, can you unleash these models on like 1 billion RNA sequences and have them search for like pseudoknots and elaborate, complex three-dimensional structures. And we will hopefully, if we find some of those from these models, we'll actually take them and try to solve those structures by cryo-EM. And so at that point, we'll know actually what level of performance you need to get to before you can discover new RNA structures. I don't have the exact number yet for the Ribonanza data set. Actually, I do have an exact number, but I'm not allowed to reveal it.
Okay.
So I can because it's based on inspecting some of the models that are coming in. So maybe we should wait until December to have that conversation. I should say I'm giving a talk at the NeurIPS conference, which is a machine learning conference. There's a structural biology workshop there, and so that'll be on December, I think, 14th-ish, around then, December 10th to December 14th. And so we'll be announcing the results there in the context of a conference. If any of you are going there, then look for the RNA talk there.
Thanks. Yeah.
There is another one from Pranav. Can existing secondary structure predictions be used for predicting the DMS and shape values?
Yeah, that's a good question. Can we use existing models to predict the DMS shape values? So I think so we have taken for the Ribonanza data set, we've taken these base pair probability predictions from a model that's called Eterna, which is a secondary structure predictor that was actually trained on prior Eterna data. It certainly does better than a baseline of zeros, but I can tell you it's not as good as kind of the models that are doing well in the leaderboard now. So it's an interesting choice. Like, do you want to use these features from previous models, which will almost certainly give you a boost when you're going into looking at the Kaggle data sets? Or do you want to avoid that bias? And I don't know the answer, but I suspect that what's happening right now in Kaggle is that, like a quarter of the people on Kaggle are trying to use some of these features, and 3/4 are not. And so we have that experiment basically going on already in Kaggle. And based on whichever models end up making it to the top 10 or the top 7, we're giving out prizes for the top 7, we will know whether or not those features will be useful. I don't know the answer.
Any more questions?
I have a question.
Okay, Helena.
Can I speak?
Yeah.
Yeah, go ahead.
In your process of going from all the chemical modification data to the 2D structures and the 3D structures, there must be you are using some prediction models for that, correct?
Yeah, so historically, like for RNA puzzles, we would use some of these Rosetta fragment assembly-based methods to.
How about you predict the 2D first?
Oh yeah. So we have a very simple, actually it's a heuristic, where we get these mutated map data, and when we make a mutation, suppose that some other base becomes reactive, we calculate a z-score, because sometimes there are just some nucleotides that become reactive or not reactive whenever you make any mutation. So we calculate a z-score, which is how unusual is it to see position 40 become exposed. And so that's a number like in, a z-score of 1 or 2 or 0 or negative. And then we use that as a pseudo energy bonus in secondary structural modeling algorithms. And so historically, we've used a package called ShapeNots that was co-developed by Dave Matthews and Kevin Weeks to now include these extra two-dimensional bonuses.
Right. So, but I guess the idea is that you use a prediction algorithm in which you inject information about shape data, DMS data, or a combination of all of them.
That's what we used to do. Yeah. But that's not, remember, for this data set, we're not providing the output of ShapeNots from these kinds of data. And actually, the reason is interesting. We were thinking about doing that because that would make some of us who are developing structure prediction algorithms, it's more convenient if we have a training data set where we have sequence structure pairs, right? But it actually is computationally too expensive for us to take the chemical mapping data and run ShapeNots on 1 million RNAs.
Okay.
It's interesting, like things have flipped, right? And so I think that's generally going to be the case. We also reached out to maybe some of the folks who are here on the call about running something like RNA Composer, but it ends up being a big computational lift to run even a relatively fast 3D modeling algorithm on 1 million sequences. And so I think the philosophy here is that's one of the nice things about getting to 1 million sequences. It's not nice if you're a human being, who's used to making models like me, but it's kind of nice because it forces you to develop methods that can scale to 1 million sequences. So I think if there's any kind of success out of this Ribonanza competition, it's almost certainly going to result in a differentiable fast model for RNA structure. And it won't require things like the Rosetta method, which is something that I helped develop over the last 15 years, which is extremely computationally expensive. And, like we could run it on maybe 10 of the sequences, but definitely not 1 million.
Okay, thank you.
Yeah.
I have a question.
Go ahead, Stephen.
Rhiju, so can you elaborate a little bit? I got a little bit lost on what the composition of the library is. So it's like, are most of this sequence or most of these sequences coming from the OpenNOT challenge? So
yeah, the way it's a plurality. So there's many sources of sequences. I've mentioned RFAM. Some of the sequences are, we take an existing sequence like the tetramer ribozyme, we make every single mutant of it, and also a lot of double mutants. They're chunks of viruses. The biggest source, so I think about 30% of the sequences in the training data and test data are from OpenNOT.
Okay, thank you.
Yeah.
Have you thought of like a scaling? Can Mutate-and-Map be scaled to that?
Yeah, so actually a big chunk of the RNA of the sequences are, we just explicitly synthesize all the single mutants of the RNAs.
Oh, I see. So it's okay, got it.
We actually took 2,000 sequences from an early stage of the OpenNOT Eterna challenge, and then we made every single mutant of those. So we have the Mutate-and-Map data for those in the training set or coming out in the test set.
That makes sense. Thank you.
Yeah. We also have double mutant data, so I didn't talk about this, but a really powerful way to check if there's a base pair in an RNA experimentally is to make a mutation to the base pair and then to one side of the base pair, make a mutation to the other side of the base pair, see if those disrupt the structure, and then actually make the compensatory mutation where a GC gets flipped back to a CG, and then you see whether the chemical mapping data are restored. So we also have a bunch of those data, those sequences in the test set, not the training set.
Okay, so maybe one more for Chai and Tanya. This time I tried to use your first name. Sorry for that. It's not always
It's all
easy.
right. Yeah, my question is related to the previous one. So a lot of the sequences are from Eterna, right? And these are design sequences. Do you think the distribution of these design sequences might be very different from like natural RNA sequences? And you know, at least in the world of protein sequences, that may mean that you may not have multiple sequence alignments. Do you think we'd have similar issues here with not having MSAs for these?
That's a great question. Yeah, I mean, okay, well, the quick thing is, you know, for natural sequences, we have a lot of natural sequences too. So I think, again, you know, when you can pick and choose a million sequences, we just include probably, I don't know, probably on the order of 30. Rachel, you might be able to tell me. Is it something like another 30-ish% of the data are from different natural sources?
Probably more. I'd say like a quarter is viral, and then like RFAM and PDB add up a lot, which is mostly natural as well.
Yeah. So
Probably
those
net
actually
a
quite a
third.
few. In some ways, so Eterna is the largest single source of data, but then there's a bunch of other separate sources that are like 10%, 10%, 25%, 10%. And many of those are drawn from natural sequences. In terms of multiple sequence alignments, yeah, it's a kind of interesting question. I mean, my personal feeling is that we're going to need an algorithm that doesn't rely on multiple sequence alignments, because for most of the RNAs that I'm interested in, it's hard to get a multiple sequence alignment for whatever reason. Those are both natural sequences. I mean, I don't know how many RNA puzzles I've done where you get the sequence and then you look for homologs and you find either nothing or you find like 10 sequences which are identical. Like, so there's no interesting signal. But then also we're very interested in, and I think the field as a whole is interested in designing novel RNAs. So you kind of really don't have an MSA.
Yeah. If I can ask a quick follow-up on that, right?
Sure.
For you guys, what would the ideal model look like? You know, so my experience with Kaggle has been that people use a lot of ensembles, and there are a lot of tricks to win these contests, right? But you know, what would be a nice clean pipeline that you'd ideally have, or does it not matter as long as you have a black box, which is really good?
Yeah, I think a black box is fine. We would like to be able to retrain it because we can get more data. So I think that's important, and we'd like to be able to fine tune it. So both of those ideally we'd have a model that's differentiable and where we can actually get the gradient. So that's why I think, but just by having the code being open source and by forcing predictions on 1 million RNAs, we're likely going to get a neural model, which will then be differentiable. In terms of how it works, yeah, so in the last Kaggle competition that we ran, the open vaccine competition, many but not all of the top predictors use something called, well, they called it pseudo-labeling or a kind of semi-supervised learning, where they used 10 preliminary models to make predictions for the test set. They averaged those predictions and they made those so-called pseudo-labels. And then they trained another set of models, and that was an iterative process. So basically at every stage, you're asking a bunch of, probably not so great models to, what is their consensus for what this important test set is? And it doesn't seem to scale very well, and it's very specific. Seems like it's very specific to the Kaggle competition. But at the same time, we'd be totally willing to do that. If we're trying to go and look through 1 billion RNAs in nature to see which ones have interesting sequences, we'd be very happy to run the top 7 Kaggle models for all the sequences and then average their results and then retrain them and then run them again. That'd be totally fine. Yeah. So I don't think there's any other kind of constraint there except speed and differentiability.
Nice. Yeah. Thanks for the responses.
Maybe I will ask one from the chat. Some RNAs have one sequence, but multiple structures. How do you deal with these RNAs with your method?
Yeah, so for the chemical mapping method, you just get for free. You get a linear combination of, effectively a linear combination of the profiles from each of the structures. So it's very sensitive to there being multiple structures, the data are. And in fact, if you make all the single mutations and do this mutated-and-map approach, there's a characteristic pattern that shows up when you have RNAs that spend 30% of the time in one structure, 70% in another structure. And so there's some papers that we published on that. And so, yeah, so a good Kaggle model needs to be able to take into account the fact that an RNA has multiple structures. And so then there's a question of, do we, I've thought for a long time that a good model, even for these data, would at some point have a built-in assumption or inductive bias that RNAs form multiple structures. Many of the existing neural nets for predicting RNA assume that there's going to be a single structure output. So I've sort of assumed for the last year that a winning model will have to at some point produce not one structure, but 10 three-dimensional structures or 10 secondary structures or 100, and then it'll compare those to the data. But I'm not actually sure that's required. And we certainly, for Kaggle, we haven't put anywhere in the discussion threads that you might consider, that your model should output multiple structures stochastically. So hopefully some competitors are trying that kind of thing, explicit modeling of ensembles. But some other competitors, maybe they'll do better just by I don't know, using some kind of a transformer and that knows something about ensembles already, or can be trained to learn something.